Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello students, a very warm welcome to the TV learning program. As usual, I'm teacher Herbert, who is going to be taking you through the biology of Year 3, a continuation of Unit 7, which is concerning about digestion in the duodenum and the digestion in the ileum. So the lesson this is biology and health science. Year 3, which is Unit 7, and which is concerning about digestion and the duodenum, digestion in duodenum and ileum. So let us first remind ourselves that is the lesson of today, that we're going to be discussing about digestion in the duodenum and digestion in the ileum. As you know, last time we discussed about digestion in the stomach. I think we can make a small review of what we discussed. We said as food enters into, as you chew the food is chewed in the mouth, and after being chewed, it is going to be broken down into smaller pieces. And now you swallow the food is going to pass through the esophagus, then it goes into the stomach, where the food is going to be churned and mixed with the gastric juice. This gastric juice, we mentioned some of the examples of the gastric juice. We talked about the hydrochloric acid. We talked some of the protein digestive enzymes. I think you remember, students. We talked about renin. We talked about renin, and we talked about the pepsin, and also tried to mention some of the functions of the pepsin and renin, what they do, and hydrochloric acid. So today we're going to talk about the digestion in the duodenum. So you talk about the duodenum, I can say the duodenum is the first part of the small intestine. The small intestine, I can say now duodenum is the first part of the small intestines, where there is further chemical breakdown of food. We say the small intestine is made up of 3 major parts. The first one is the duodenum. The first one is the duodenum. The second one is the duodenum. And the third, which is the ileum. So these are the 3 major parts that makes up the small intestines. The small intestines. You say in the duodenum, so we're going to discuss about digestion in the duodenum. We talk about digestion in the ileum. Of course, when you talk about when the food is churned, is being broken down in the small intestine, the food has to be released into a sphincter muscle, which is called the pyloric sphincter, and it goes into the duodenum. And the duodenum is the first part of the small intestine, where further breakdown of food takes place. If I'm trying to show you, this is the diagram that tries to show the structure of the stomach. And you can see in the stomach, we have what you call the first part of the small, this is the stomach, the first part of the small intestine, as I can try to figure it out, which is like that. We try to figure it out, it's smaller part of the small intestines. If I'm trying to draw it very well, all right, this is the part which is accessory organ. We have that part. Let me try to draw it very clearly. We have the diagram which is drawn like that. This is the stomach, and the stomach, from there we have the first part, which is the duodenum, and we have the part which is the liver, and this is the pancreas. From the pancreas, we have another part. This is what you call so from there, we have the first part of the small intestine, which is the duodenum. I can say this is the duodenum, all right. And this is the pancreas, as I can try to figure it out. This one is esophagus. Now this is the stomach wall. This is the pancreas. This is the liver. And this one is the gallbladder. The gallbladder. You can say the gallbladder, or in bracket you can say GB, the gallbladder. So the gallbladder produces what you call bile. And I can say, this is to say, you see, this is a small diagram that tries to show. So the first part of the small intestine is the duodenum. So what does the duodenum do? The duodenum, as I said, after the food is churned, the food is going to be changed into a semi-liquid substance, a same liquid, which is a greenish in color, a liquid substance called chyme. Now, as you can see, after food is being broken down, the food is going to be raised into the duodenum. What is going to be found in the stomach, it is called chyme. The chyme is a liquid substance, same liquid substance that is in greenish in color. And that's liquefied substance, which is called chyme, is going to be raised into the duodenum for further being broken down. Now, after reaching into the duodenum, we know the chyme is the environment in the stomach, which is going to be raised, the pH in the stomach, is ranging between 1.5. Now, if that chyme is going to be raised into the duodenum, it means it has to be neutralized. And there must be, some of the substance has to be raised by the pancreas and even the liver, as I mentioned. So what are these enzymes that are going to be raised by the pancreas? Pancreatic enzymes that are going to be raised. Pancreatic enzymes that are going to be raised to act upon, to break down the food, further breakdown of the food. These enzymes, we have amylase, the amylase. If I say amylase, I mean pancreatic amylase, which breaks down starch. You can say breaks down starch. We have another one which is the protease enzyme, which breaks down fats. So these are pancreatic enzymes. So this is number 1. This is number 2. We have number 3, an enzyme which is called lipase, which is also going to be raised by the duodenum. The lipase, which breaks down. Protease breaks down. Sorry, this protease breaks down proteins into peptides. Sorry, and then the lipase breaks down fats. You also have what you call another enzyme, which is called nuclease. The nuclease enzyme, which breaks down nucleic acids. So, but there are also some substances that are going to be raised by the pancreas. What we call these substances, other substances that are raised by the pancreas, other substances that are raised by the pancreas to perform the function of breaking down the food is the bicarbonate. This bicarbonate is very important because, as I told you, this once the chyme is going to be raised from the stomach, it is going to be raised with a pH which is ranging from 1.5. So once it is raised, the pancreas has to raise. I can try to show you. Let me just pieces of chalk. So I'm saying that the pancreas is going to raise This is the diagram showing the pancreas is going to release some enzymes and also release bicarbonate, which is very important. I said bicarbonate is important. It helps to neutralizes the acid from the stomach, to neutralize the acid from the stomach. So it means that once that chyme is going to be released with high pH, which is less pH, which is ranging from 1.5, it has to be neutralized by bicarbonate released by the pancreas, so that once that chyme is going to be released into the duodenum, will not be able to affect the walls of the small intestines. I mean the microvilli, the villi, and etc. So these walls cannot be affected very much. So we can say these are some of the enzymes that are going to be released by the pancreas and which facilitates the breakdown of the food in the duodenum. I can say the chyme is going to enter into the duodenum, and I say the pH in the duodenum is 1.5, which needs to be neutralized because the environment in the duodenum is an alkaline environment. So it has to be neutralized by the bicarbonate, which is released by the pancreas. So we say, I mentioned the number of the enzymes, and we can say very much this bicarbonate is very important in neutralizing the acid from the stomach. Let's talk about now what are the functions of the bile. The river contains bile. Contains the gallbladders. Sorry, it contains the gallbladder. The gallbladder. The gallbladder is very important to release what you call bile. It releases the bile and bile pigments and even bile salts. The gallbladder releases the bile, and one of the functions of bile is emulsification of fats. The function of bile is emulsification. If I say emulsification, it means breaking down large droplets of fats into small droplets of fats so that they can just be able to be dissolved in water. So it makes fats to be dissolved, because once these fats are going to be released, are found in a chyme, once they are released, they have to be able to be broken down into small molecules so that can just be able to dissolve in water, and that one is normally done by the gallbladder that releases the bile. The GB, which is the gallbladder, releases the bile, and bile plays a very important function for emulsification of fats. So that is the digestion in your duodenum in simple understanding. You can say once the food is released from the stomach, which is a chyme, the chyme is going to be released into the duodenum. Once the duodenum detects the food, the chyme is coming to the duodenum is going to be stimulated by some of the hormones, which are called secretin, which is going to stimulate the pancreas to release pancreatic enzymes and also to release the bicarbonate, and also stimulates the liver to inform the gallbladder to release the bile. And I said bile is very important in emulsification of fats. So let's talk about digestion in the ileum. Digestion in the ileum. So simply, I can summarize it in that way. You say the duodenum is the first part of the small intestine, and you can say the small intestine is made up of three major parts: the duodenum, the duodenum, and the ileum. I can say the first one is the duodenum, the second is duodenum, the third is the ileum. I said the digestion in the duodenum starts with from the stomach. The food is going to be churned after mixing it with the gastric juice. It is going to be released as in form of a liquefied substance, which is called chyme. The chyme is going to be released into the duodenum, and once it is released into the duodenum, it stimulates the walls of the duodenum to secrete some of the hormone, which is called secretin hormone, which stimulates the pancreas to release pancreatic enzymes. These pancreatic enzymes, I mentioned them. We have the amylase, pancreatic amylase, which breaks starch. We have protease, which breaks proteins. We have the lipase, which breaks fats. And we also have bicarbonate, which is also released by the pancreas, which is very important to neutralize the pH of the chyme, which is 1.5, to a neutral pH, which is almost like, which is 7, which is neutral, so that the wall of the duodenum cannot be affected by high pH, And also talked about the gallbladder. The gallbladder produces, the GB produces what you call bile, and bile is very important to do the function of emulsification. If I say emulsification, that is breaking large droplets of fats into smaller droplets of fats so that they can just be dissolved into water. Now, let's talk about digestion in the ileum now. If I say ileum, digestion in the ileum. Digestion in the ileum. Students, if I say digestion in the ileum, let's have the diagram here. I will try to have the diagram, and I try to show you where the ileum is located. I said this is trying to show you the structure of the alimentary canal. We have the mouth, stomach. I said the first part of the small intestine, we have the duodenum. We have the pancreas. I said the pancreas is very important because the pancreas and the liver, we call them what we call accessory organs. The pancreas produces pancreatic enzymes, as well as the liver produces bile. And bile is very important as far as breaking down large droplets of fat into small droplets of fat. We have now the small intestines. The small intestine, we have the last part, which is called the ileum. I can say in the ileum, that's where the absorption takes place, all right? I can say, let's first think about what are the functions of ileum, students. Think about what are the functions of ileum. What are the functions of ileum? Students, we say, what are the functions of ileum? What are the functions of ileum, which is the third part of the small intestine? One of the functions of ileum, think about it, what are the functions of ileum as a part of the small intestine? One of the functions of ileum is to complete the process of absorption. Completion of the process of absorption is normally done in the duodenum. That's supposed to be the complete breakdown of food is normally done in the ileum. We can say complete breakdown, you can say complete the process of breaking down. Let me just use that word, which makes sense. You can say completes the breakdown of food. Number two, we can say the duodenum, another function of the duodenum, it is a site for absorption. The site of absorption is found in the duodenum, in the ileum, sorry. It is found in the ileum. So ileum, it is one of the functions of the ileum, it is to complete the breakdown of food. The second function of the ileum is the site for absorption. We normally know that the ileum is a site for absorption. And we say in the lining of the small intestine, we have what we call epithelial lining, which normally have what we call special cells. These special cells are called the goblet cells, which normally secretes the mucus, And I say this mucus is very important to protect the wall of the stomach not being attacked by hydrochloric acid. All right. So write the function of it. Take that note. Let's have, I said that if I can try to mention it, I said the epithelial lining of the small intestine has a special cells. These special cells are called goblet cells. The goblet cells that secretes mucus, and this mucus protects the small intestines from being digested. And I say being digested by what? This is being digested by digestive enzymes. And also, it also have what you call secretory cells. These secretory cells are found in the intestinal walls. Also, which produce what you call intestinal juice, which is called secus entericus, which is very important to bring what you call a slightly alkaline medium, so that the wall of the small intestines cannot be affected by hydrochloric acid or by the chyme, which contains high pH of acid. Quickly, let's talk about absorption in the small intestine, right? Absorption of digested food in the ileum. Students, we're going to discuss about how does the absorption of food, the absorption of digested food takes place in the ileum. We say I mentioned that one of the functions of the ileum, it is complete breakdown of food. Second, we said it is a site for absorption. So we can say most of the absorption of digested food takes place in the ileum. It's because the ileum has got some of specialized structures that enables it to carry its function. What are those adaptations and characteristics that makes the ileum to perform its function? There are a number of adaptations. Before we talk about it, you can see, try to check on the diagram. This is the intestine villi. And we normally try to allocate that in the duodenum, there are also some villi. All right? They are in the ileum, there are some villi. In the ileum, there are villi. They are called finger-like projections. These finger-like projections, they are very important to facilitate for absorption of food. Let me just bring some example. The villi are found in the ileum. Some of you bathe. After bathing, you try to use a towel to remove water from your body. So in the small intestine, there are structures which are called the villi. These villi, they are very important to absorb the digested food and carry them to the bloodstream, where they're going to carry their function. I have a diagram here. The diagram, let me just try to show you the diagram, which is very clear, and you can understand it very clearly. I have a diagram. The diagram here, you can try to see, this is the wall of the small intestine. And in the small intestine, we have this one with the vessel with a blue color. We have another one with a red color. This means that one carries oxygenated blood and supply to the small intestine. And we also have this one, which normally carries the oxygenated blood, that I mean carbon dioxide and other waste products from the small intestine. We have these finger-like structures, which are called the villi. When it is one, it is called the villus. So in our small intestine, there are millions of the villi. And these millions of villi, which are finger-like structures, they help the process of absorption. And we can try to see on the villi, you also have a microvilli. These microvilli, they also play a role in absorption. What are these for? These villi, finger-like structures, they are very important to perform the function of absorption. We have another diagram which is trying to show you a detailed structure showing a villus. This one is a villus. This one is a villus. As you can try to see, this is a villus. The villus is made up of very many parts. We have what you call epithelial cells. We have blood network capillaries. We have these epithelial cells. You also have what you call mucus secreting cells. These are mucus secreting cells. Mucus secreting cells. We have lymph vessels. We have arterioles. We have the venules, and etc. So this is the internal structure of the villi. And I said the villi is normally found in a small intestine, in the ileum precisely, I mean in the ileum, which plays a big role in absorption of the end product of digestion. Let's talk about what are the adaptations that enables the viri to perform its function. The viri must be having characteristics and also adaptation that enables it to carry its function. Here I have one of the characteristics of one of the adaptations of the iridium to its function. Number one, we have large surface area over which absorption can take place. This is created by a long length of the iridium and a highly folded of its inner wall. The inner wall is folded. As you try to see, the inner wall is folded, This one you can even see on the diagram, the inner wall is folded. So that one creates a large surface, creates a long length of the iridium and a high folding of its iridium enables it to carry a process of absorption. We have what we call thin epithelium. This one is called epithelium cells. Thin epithelium. On the diagram here, we have what we call epithelium cells. Thin epithelium. The thinner the membrane, the faster the rate of diffusion. Thin epithelium through which digested food materials diffuses into the blood. Number three, it has very many blood capillaries, or a dense network of blood capillaries. So as there are many network of blood capillaries, that one facilitates a rapid transport from small intestines, the digested food materials that diffuses through the epithelium. So number four, they have finger-like projections. As you try to see this diagram and you see it with my finger, they are finger-like projections. These finger-like projections are called viri. These viri, as you can see, this is a structure. This one has been extracted in a small intestine, which are called the viri. These finger-like projections in senior are called to be villus, which increases the surface area of iridium for absorption of digested food. The last one, it possesses lactile vessels, which is very important in absorption and transportation of fats. So that is the end of the lesson. Let me just give you a homework exercise. Give at least 4 adaptations of, that is number one, of iridium to its function. Number 2, give and explain the enzymes produced by the pancreas. Number 3, you give what is the function of bile. So students, thank you very much for being with me in my lesson. We will meet next time. Thank you very much.